Good afternoon, everyone. As Dr. Zivadinov brought up, we have a lot of facts, but still a lot of questions to answer for. And I would like to have, during my next slides, going over the specific steps and rules for a scientific hypothesis to be proven. The question is, if any hypothesis scientific exists for two factors, one is the cause for another event, we have to prove, and the easiest ways to do, in a one-time event, to prove that this factor is associated with the other one. In our case, CCSVI with MS. Is CCSVI a higher number seen in MS patient? Is this indeed increasing the risk to develop MS? As mentioned before, we were trying, and we tried before, and this was also will be the future for, how we organize and design our studies to provide an answer. So, as already mentioned before, the best and the real proven will be a prospective studies. However, this takes a long time. MS is a very long, right, disease, chronic disease. There's so much difference in between patients, heterogeneity. We want to have a first initial answer. One time, evaluate the patient. How can we get the most information faster? So this is called what we call one-time cross-sectional observational studies. What we have to do, try to get and evaluate as many MS patients we have, different diseases, different ethnicities, different disease stages, very early on, late on, and determine what is the difference. We have to do in the same time healthy controls. Is this element seen also in healthy controls with no relation to MS, right? And we have to have other diseases. MS may overlap with other diseases. So in order to learn this, we have to go and look on a very large population. The numbers is still not known. And this is why, as we'll show you before, we initially decided on 500 volunteers, but we'll go further. For 1,000 patients, it's our goal to get a more explicit number for one time, what we're calling cross-sectional data. And again, looking for the association. The second, the most important, if two factors exist, is this indeed a cause for the disease? That is much more difficult. And I will have you showing after then a slide that goes step by step, provided by statisticians. They are kind of, running our show. But that's very important. Rules to try to prove, is indeed this factor cause and effect proven? It is more difficult, but I will show you what we have up to now and where we're going and targeting to prove that this is the cause. Clearly, for the cause, one of the elements that can help on it is intervention, right? We have an abnormality, let's fix it, let's see how it is going. It's not so simple, but we did already start on this, and Dr. Siddiqui will elaborate more what it was done, what we did before already, and what we're looking for further on our patients, and also in other models, virtual models, video, or animal models, right? So altogether, as I said, now we're trying to figure out, is this association, CCSVI, or venous abnormality, a higher risk for someone who has it to have MS? So what will be next? What do we have today? We do know today from our study, but from other studies too, that many healthy people have CCSVI. We have 20%, but they are. We do have too that MS patients do have CCSVI, and indeed in a higher percentage than the healthy controls. However, we do have MS patients without CCSVI, right? So what this means is that CCSVI, the presence of this hemodynamic abnormality, are not necessary to have MS, because we have MS patients with no CCSVI, but at the same time is not sufficient because we have CCSVI and there's subject to not have MS, right? However, again, we do have the clear information, at least based on our data up to now, that MS patients do have more frequent CCSVI than healthy population. And we also did see, at least in our initial group, that more progressive patients do have a higher frequency with CCSVI up to 90% versus the very initial patient that had the first event. We even do not call this MS, we're calling this clinical isolated syndrome, and even before we're calling this radiological isolated syndrome. So no clinical presentation, only the MRI shows MS. So with this data, evaluating, we try to figure out indeed what is the role of CCSVI on the risk for MS, on the risk for propagation for MS. What is the hypothesis, actually, that was brought up primarily by Professor Zamboni as linking, right, venous problems and MS? It's very important, right? So what he brought up, and probably there is a certain, basis to it, is that if we have these hemodynamic problems in which the blood is refluxing, going backwards in the brain instead to go down, it may, be associated further with stasis. The blood stays there. And what's happening? It may happen, as we often, he very much, Professor Zamboni is a venous specialist. And he's primarily comparing with what's happened with the venous problems when we have in the lower extremities with the venous insufficiency. And clearly what he hypothesized, based often on what he saw in peripheral problems, is that we have a venous stasis. The blood cells, primarily the red cells, the erythrocytes, can leave the flow and go into the tissue around. The red cells have a lot of iron, and they may deposit around in the tissue. This is why when we have the venous insufficiency in the legs, right, varicose veins, we have this changing color in our area. The same thing, this is primarily iron deposition. We have a certain possibility, our tissue is able to, fix it, but if the certain amount is too much, we're going over the possibility of cleaning up. And then it becomes toxic to the tissue. So what the next? The cells and all our, immune system try to help to clean up, but may be activated. And an MS patient that already has an active immune system may go over the board. And then it may develop all the big cascade of pathological abnormalities associated with MS. This could be a theory, it could be much more, and we're trying to figure out and understand better. Now I told you about the cause. What are the real steps and usually required to try to prove cause-effect? And it was actually statements done by Bradford Hill, in which he took as a statistician from Britain, that he took as a model smoking, lung disease, and cancer. As we all know, this is well accepted. Smoking is associated, right, with lung problems, emphysema, and cancer. However, we all know that maybe patient with cancer never smoked, and we know that people smoking, not everyone has cancer, right? But the causality is well accepted on it. So we're here where we are. Now, what is he considering the important steps to be fulfilled in order to show cause and effect? First, a strength association, meaning the number of the cause should be along with a high number of the effect, meaning a lot of smokers and a lot of cancer, for example. Second, consistent. One study has to be supported data with the second study. So we have consistent data coming from all the studies. Second, specificity. Is one cause provided a certain effect? We don't want one providing multiple. It could be multiple, but it's primary one effect. Second, coherence. Is this association really fulfilling all what we know about this disease and relation? Meaning it doesn't have, we try to be, going along and pursuing what we know already and consistent. Temporality, this is a problem. Meaning try to have, if you cause and effect, clearly the cause has to come before the effect, right? And this is something different for a disease that is so long time. And sometimes we have an A, the cause, and we have an effect, C. You have something B in between. And so the A is going to B, and B is going to C. So we don't have a clear-cut direct between A and C. So they are pretty much difficult to identify this temporality. Second, exposure response. If indeed the factor, the cause, is providing the effect, we have as longer time you have exposed to, as stronger the cause is, you should have clear-cut, much higher chance to have the effect and maybe worse the effect, right? Biological plausibility, what we talked about, we have to go along with what we, pathobiology explain. This is going this because physiologically this is usually how we know from all anatomy, biology, and so on. Experiment. Can we intervene and stop, improve the disease by terminating the cause? That would be the great, again, in human, but also maybe we should consider more also in animal models and virtual models to do. Is any analogy, meaning is this presence of certain cause doing something else? It's pretty similar, maybe in different system and so. And I'll show you, further possible analogy what CCSVI can do, not only in MS patients.